Back to the lesson 3 tutorial for functional annotation, where we're going to be talking about and using the tool Hammer to do a homology search using position-sensitive models to determine our functionality. So here we are in the, if you follow the link in the video, we're in our topic functional annotation repo as part of the BVCN, and we're in lesson 3, the directory. The files for that presentation that's associated with this tutorial are available. And this is the README that we're going to do to follow along working with the tool Hammer. And so I have launched already my binder. You can do so by clicking on this particular badge. It may take a minute or two, depending on when the last time this was used by somebody, but do know that it works and it will load up. And you can ignore all of the errors that pop up, and you'll get to a point where you can then follow along with the tutorial. So today we're going to be talking about Hammer, and specifically we're going to be looking at a Bryozoan, wow, it's been a long day, Bugula neratina. And so I'm a microbiologist, and every word that I say from eukaryotes always seems like I'm saying it wrong, so hopefully that checks off as being useful. So it's an old invertebrate, millions of years old on the tree of life. And this is what they look like, and they can colonize rocks in the marine environment. And so what we're going to do is we're going to use a recently sequenced genome for the Bugula neratina to look for specifically a subset of proteins that have to do with innate immune response. And so it's a signal transduction protein that we're going to look at with the name MYD88. I will probably end up calling it a bunch of times MyD88 just because that's what it looks like, and isn't that just more fun? And so there's a nice rundown here written by one of our instructors, Adelaide Rhodes, who will give you some overview of how this particular protein works in terms of its way of triggering a response within the host based off of the way that it interacts with its symbionts. There's some additional information, including how we got the protein sequences down here. And I just want to mention that we pulled them all from Uniprot, and how to do that is actually going to come up in a later tutorial, so we didn't want to spend time looking at it, as well as a paper that describes how the MyD88 immunity gene functions within organisms. So some really nice extensive background on why we'd be interested in these proteins to begin with. Okay, and with that, what we're going to do is we're going to transition over into our tutorial, and we're going to do that by launching our terminal. So as part of this lesson, what we're going to do is we're going to compare our MyD88 proteins against the Bugula neratina genome. All right, and so what we're going to do is once we've loaded up our binder and we've entered into our terminal, we can, I'm going to start up here. Okay, we're outside of our particular database, our data set that we're going to work with. We're just going to cd into our directory example data. And so that's going to pop up. There are two files that we're going to be working with. So one is a set of 16 MyD88 proteins, which are in the MyD88.faa file, and then a canoe filtered Bugula neratina proteome, which we have in bugula.pep. All right. So if you were working with us in lesson 2, what we want to do is we're going to compare our BLAST results with our Hammer results. And so if you were working with us in lesson 2, our first step to working with this is that we have to make a BLAST database. And so we're going to do that really quickly. And so what we're going to do is we're going to read in our peptides sequences, and we're going to create a database type that is protein. And that's going to run. It's going to take a second or two. And then what we're going to do is we're going to have created a set of binary files that are our proteins that we're looking at in compressed space that help BLAST run a little faster. And then we're going to do our BLAST P search, where we're going to use our query, which is our MyD88 sequences, our database, which is the Bugula peptides. We're going to use out format 6. If that means nothing to you, you should definitely check out lesson 2. We're going to set an E value of E-5, and then we're going to output it to a file that we're going to call blastp.myD88 pep out format 6. And so I want to take a second here to really emphasize this output file name, because I think it's probably one of the best examples I've seen of this in a tutorial. And so this tutorial also comes from Adelaide, which is very nice of her to share this with us. And so what you can do with these types of files, because especially when you're doing this in time, and especially when you're doing it in this kind of methodology where you're looking to generate models and do searches that have and figure out where the meaning is on certain cutoffs, labeling it to a very high level allows you to come back at some point weeks or months later and know what you were doing. And especially if you start to standardize that early in how you name things, it's really good. So this tells you the program that you ran, what your query was, what your database was, what format it was in, and then the output format that it's also in, right? So without opening the file, I know exactly what's in there, which is particularly fantastic, right? And you'll see that as we name our files going forward. And it definitely is a skill to be developed. And so what we want to do is we want to check out what's in our contents from our BLAST P-search using less. And what we'll find is that there are no proteins that are similar to the proteins related to the MyD88 protein based off of BLAST. So that's it. We're going to wrap it up here, and that's going to be fine. Obviously, that's not the case. We're actually going to keep moving forward, and that's intended. So what we're saying here is that when we use BLAST to compare for primary sequence similarity, looking for homology for the sequences, we don't get a match in the Bugula genome. Okay, so the reason we're doing this is because if we take these sequences and instead of looking at them as, well, what are the similarities between this and the organism that we're looking at, and we converted this into, well, what are the important features of this protein before we go and do our search? Are there conserved features that we can use to identify this, right? We can use something like Hammer to set this up, right? And so we have to get to a point where we have a hidden Markov model. And so there are a couple ways of doing that. And one is going and finding a pre-computed HMM profile from a public database like PFAM, or we can create our own, and that's what we're going to do here. And so this is a simplified version of what we'll do in later lessons. But what we need to do is we need to take our sequences, which are in our FASTA format, and we need to make an alignment, a multiple sequence alignment. And so we can do that with muscle. And so we're not going to get into muscle too much in detail in this particular lesson, but maybe in the future. And so what we're going to do here is we're just going to call it MSA for multiple sequence alignment. We're going to hit enter to run it. And because there's only 16 sequences, it actually runs fairly quickly. And so then we can actually look inside that sequence to see how this looks, or inside this file to see how it looks, to see how it compares. And so we've converted our normal protein sequences, which just have either one line or 80 characters and then a return character. And we've converted it into an alignment. And we can see that based off of these gaps that we're seeing here. And so one of the things that you can see, or you might notice if you've looked at alignments like this before, is that there are some regions that seem to not be ideal for this particular setup. And this can be somewhat debatable, right? So we have a situation where there's a lot of elements in the middle of the protein that seem to be, at least to be aligned, right? Sure, there are variations in the gaps, but for the most part, we have sequences that are aligned. But there are also two big regions that seem to be pretty common throughout the alignment, which almost exclusively have gaps in the sequences that we're looking at, right? So both here, gaps, and here, gaps. And so if we were to scroll through this just by pushing down, we could look for proteins that maybe have sequence there where none of the other proteins do. So we're going to keep going down. Here's our candidate for the protein that has sequences at the end of its sequence that the other proteins that we're looking at don't have. Maybe this also then aligns up here. So maybe it's not universally not something that's there, but it's something that we can think about. And then going down some more, we can find the protein that then has sequences almost exclusively in the early part of our protein here, where again, we have a large amount of sequence that's here that doesn't seem to be present in any of the other proteins that we're looking at as part of this alignment. And so if we wanted to, we could actually go in and we could curate this alignment a little bit further. And so the first thing I'm gonna suggest we're gonna do, you don't always have to do this, is I would suggest that we delete our current alignment because we're probably gonna end up not using that going forward. So I'm just gonna do that and do remove my D88 MSA. And then we're gonna use the screen editor, text editor called Nano to bring this up. And what we're gonna do is we're gonna process down until we find the names of these particular genes that we're looking at, and we're gonna remove them. And we'll do them, it's pretty easy to do this for a couple of genes. I would not recommend doing it this way for a large data set or large sequences that you wanna look at lots of elements of the alignment. It just happened to work really nicely with my D88 sequences. And we can use some of Nano's built-in functionality to actually do this pretty quickly. So one of the elements that we want to search off of, and this seems to be pretty convenient right now, is this, we can use this sequence name ID. So this M4I212 Craggy, which actually, if we look, is the first sequence in our data set. So we can remove that, and we can remove that, if we're gonna do this manually, we need to do line by line until we get to our next sequence that's down here. And so we can do that by holding Control and then hitting the key K. And so if I hit Control and hit K, I can remove line by line. I'm just cutting them out. Very similar to like any other text editor, I'm using the cut command to get rid of them. And so I've now removed all of those. And now I wanna find this other sequence, which might be somewhere very far away in the file or somewhere really close by, like the next one down. If we wanted to look for it, we could hit Control and then press the button W, which will bring up this search box, and then we can just type and figure out and use that. So I can type in A0 A to B for RVP2 and hit enter, and it takes my cursor to that particular spot. So now I can move around, and then we'll use Control K again to cut out the lines. So we'll just do boop, 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 boop. And we want to make sure that we get everything, because otherwise we'll end up with sequences that are merging. We want to make sure we don't cut the next line here, because otherwise we'll lose a header. And so we've now taken our 16 proteins and reduced them down to 14. And so to exit out of Nano, you hit Control X. It's going to ask you, do you want to save your changes like any other good text editing program will? You're going to say yes by hitting Y. And it's going to say, do you want to write it out to the same file that you were working in? And so we can make changes if we wanted to here. If we wanted to write out to the same file, we just hit enter. And then we have our file saved with less sequences in it than we did before. And so now we can rerun our muscle search. All right, so we have our newer alignment. We can look in our output to see what that looks like. So here we have, again, less gaps in the front, maybe still more in the back. We could refine this further if we wanted to. But again, what we're actually really interested in are these regions that are conserved for all of them, right? And we know that HMMs have the ability to ignore those areas that have high areas of gaps without making it so that they aren't significant in the search space. So we can use the alignment that we have right now. All right, and so we want to convert our multiple sequence alignment into an HMM profile, which we do using the tool HMM build. Where we do that so our input or our output is going to be the HMM file. Our input is my D88 MSA, and so this is an important thing of order here. It's looking for the file that it's going to output to before the file name of the input, and so we'll make sure we give it to it in the right order. We're going to hit enter. It takes a very short amount of time to run. We're going to use this HMM profile going forward. So we're going to actually convert it into a compressed binary file to allow the search speed to happen a little faster. So again, we're going to use HMM press for that. And then you target the HMM file. We're going to hit enter. And then I'm going to show what this looks like, where we are currently with our data by hitting LS. And so we have our BLAST output, we have our peptides, we have our BLAST database, we have our input sequences that we're looking for. We've converted to an HMM profile. We've made our multiple sequence alignment, and now we have 4 binary files that represent a compressed version of our HMM profile that should speed up some of the search time related to HMM scan. Okay, so that's what we're going to do. We're going to use HMM scan to search for our newly created hidden Markov model in the proteins of the Bugula genome. Okay, so let's do that. HMM scan. Again, so this is a really good example of a well-documented output file. So here we're going to use specifically a domain output. That's a table output from HMM scan. So it's going to give us back our best domain matches. And then we're going to type in the name of the file, which is going to be nice and extensive. .myd88.dom table out. And then we want to give it the profile that it's going to be looking for, which is our HMM file. And then we're going to tell it which proteins to search it against, which is our protein database. And so when I hit enter, this is going to take a little bit of while to run. It's going to run for about 1.5 to 2 minutes. And so you'll see it, and then we'll talk about what the output looks like after it's done running. So I'm going to hit enter, and it's going to run. And so this is each individual protein of our protein file being searched against an HMM, the HMM profile.
I just thought I would pipe in while this is running because it's going to take a little while about why I did this. The Bugula neratina genome is possibly 250 million years old, so it's really separate from other organisms that might even be closely related. There's lots of controversy about where it fits in the tree of invertebrates, like where does it fit in. So doing these protein searches is actually very manual. There's no easy way to get there from here because there's no similar organism that's been sequenced before. I mean, there are a lot of similar organisms. There's other bryozoans. It's just there's a lack of sequence in the databases. So this is something that's very typical for non-model organism people, that you have to kind of reach a little bit to even get an answer to a question. So my question, what was my biological question, was bryozoans have a set like symbiotic community, a microbial community that it allows to colonize. So usually, an invertebrate wants to reject pathogenic organisms. So it builds up a defense mechanism, this innate immunity to recognize things that are going to harm it or give it a disease, like a virus or a bacteria. But in many cases, just like coral and zooxanthellae, there's accommodation for certain organisms. And this is an evolutionary relationship that's built on, millions of years of association in the environment, where some animals will tolerate, some eukaryotes will tolerate these invaders, right? So it's an interesting question in general. And finding these genes is not easy. You guys are doing like part of the work that we actually did for the paper. I was just curious about how Bugula neratina might be differing from other organisms and allowing symbiotic things, which might be seen as invaders, to coexist with it. So it has to turn off, it has to recognize certain bacteria or fungi or, other things as a friendly organism versus something that might hurt it. So that was a research question we were pursuing with this inquiry. But like I said, we had to be very manual about it because there's no closely related organism which we could just blast against.
So after a couple of minutes, we now have the ability to look at this output. And so let's take a quick look at this. And so what we should see, and I'm going to make this a little bigger so we can look at it on one line, is an HMM hammer output table that demonstrates that approaching it from this manner, where we're actually looking at from conserved functions, conserved parts of the protein related to this innate immune response. Now that we've moved back, so we're not just looking for primary sequences, but we're looking for what actually matters and what is a conserved element to this. We actually see there are 3 matches between our target HMM model and the proteins in our query genome. And then to walk you through some of the outputs that come out in this table, you'll notice that these outputs aren't that dissimilar from what we get from our BLAST outputs. We get a number of scores, including how well the query matches our targets, as well as a breakdown of how these sequences compare both from the full sequence approach to the domain that we're looking at specifically. And so we can get multiple domains when we're starting to look at proteins that match up, and each of those domains might have significant values on their own right. Relative to that, you can see that there's a bit score here that functions very similarly to the bit score that we get with BLAST. E-value functions the same way as we do with BLAST. And so you can use those values as scores that you can then filter on going forward, the same way that we might use percent amino acid identity or percent nucleotide identity in a BLAST search result from a table like this. And you can notice that all of the non-numerical outputs or tabulated outputs all have pound signs, and so we can use those pound signs as a way of looking for lines that we don't want to query for our numbers. So it turns out there are actually 3 matching proteins that go along with MyD88 in the target organism that we're looking at. So one of the things I wanted to talk about, and I talked about in the lesson, is when you would want to use the difference between, or why you'd want to use some HMM scan versus HMM search. And so in our next version of this, what we're going to do is we're going to run the same process, but we're going to just change the tool, right? So it's exactly the same. We're going to use a different table output. So this is the more generic table output, not the domain-specific table output. We're going to run HMM search, table out. We're going to call it basically the same thing, except then we're going to call it table out as opposed to a domain table out. We're going to put in the HMM profile we're searching, the peptide file that we're searching, and then we're going to hit enter. And unlike HMM scan, which took somewhere on the order of about 2 minutes, you'll see that HMM search finishes very quickly. And this has to do with the fact that the HMM only has to be loaded a single time for this search. So it gets loaded once and then gets compared to all the proteins very quickly, as opposed to in the other situation where we took each protein and then had to open up the HMM file to make the comparison. And then we go to the next protein and make the comparison and go forward from there. And if we look at the output, we'll see that the output is also identical. We get 3 matches. And then here, what we've done is we've, some of the elements are different because we're using the full table output. Here, again, some of these values are different, but and more specifically, our subjects and queries have been switched here, where the proteins are now the query as opposed to the model. So yeah, so we actually have that ability to do that as well. HMM search. Okay, so we could do this going forward, and we could do something where we approach a way of trying to annotate the full genome using PFAM, which is a public database that has about 20-ish thousand HMM domains already pre-calculated. And so we're not going to do that here because PFAM is fairly large. It's a couple gigabytes. And so having the binder have that download and then run it, if we actually were to run it, it would take a little bit of time. So we wouldn't be able to do it in a reasonable amount of time. And so as we wrap up, these are things that there are some questions that we might want to consider for this going forward. Things like, how much does a reference database influence our ability to annotate a genome? And so this comes down to that fact that because of the phylogenetic position of the organism we were working with, proteins in the databases didn't have high level of similarity to it. So when we did a simple search using BLAST, the output wasn't able to detect something. But when we converted that information into something that looked at the conserved elements that make the protein what the protein is, we were then able to then use that and look at more distance relationships and look at how that sensitivity works between BLAST and HMM profiles. And so then you might want to think about this in ways that this would influence a robust annotation pipeline going forward, right? So if you're working with a pipeline and how it's determining what your annotations are, it might be very relevant to think about how often it's using something like a local alignment search to make decisions, and how often it's using something that's a little bit more sensitive like an HMM model. And so with that, we'll wrap up lesson 3, and please come back for lesson 4, where we start talking about specific databases and how those databases can be used for annotation. Thank you.